We'll be going not this Wednesday of this week, but Wednesday of next week on the field trip. And most of you have gotten those field trip permission slips back to me, but some of you have not. And so Barbara, Juan, Megan, Jessica, Ivan, Ramiro, Sandra, Julio, Christina, and Carolina. You guys are on my bad list. So get those back to me as soon as you can, please. What other announcements do I have? It's supposed to be hailing in Monterey. So I like cold, wet days like this. It's a good day to be inside learning some science because there's no sunshine where you go, gee, I'd rather be out playing on the grass or something like that. Let's see, what else? Not today in lab, but on Wednesday on lab, we're going to do a lab on proteins. And when we do that, I'm going to finish off the protein lecture as part of the pre-lab lecture. So it just means bring your protein lecture notes when you come in on Wednesday this week. Alrighty. All right. Well, I think it's been a long weekend and a couple days since we covered the nucleic acids. So let me do a real quick recap on them. Nucleic acids are one of the biological macromolecules, one of the big molecules you find inside all living things. And like all the macromolecules, they are polymers built out of smaller molecules linked together called the monomers. And the monomers of the nucleic acids are called the nucleotides. And here's the basic structure. All nucleotides have a phosphate, which remember is a phosphorus atom surrounded by 4 oxygens. And that's connected to a ribose sugar, which is a 5-carbon sugar. Here are the carbons. Well, there's carbon here, 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 and here. And you probably remember from the laboratory last week on carbohydrates, all monosaccharides have lots and lots of hydroxyl groups. There's an OH group right here, an OH group on this carbon, and this one right here, this carbon doesn't actually have a hydroxyl group, but what it's connected to is what's called a nitrogen-containing base. And we talked quite a bit about these nitrogen-containing bases. They're part of the nucleotide molecule, but they have individual names. There's one called guanine, there's one called adenine, cytosine, thymine, and another one called uracil. And you find nucleotides that can have any of these nitrogenous bases here at this location. Now, all that being said, there were 2 subvarieties of nucleic acid, nucleotides. One type is called an RNA nucleotide, and it looks pretty much like this. It has both oxygens down there and down there. But RNA nucleotides could not have one of those 5 nitrogenous bases here. Does anybody remember which one RNA nucleotides cannot have? T, right. In RNA nucleotides, they can't have T, they can have G, A, C, and U as their nitrogenous base. For DNA nucleotides, they are missing that oxygen right there, so that's where they get their name. They are deoxyribose nucleic acids, DNA. And what one can DNA nucleotides not have? U, right. So it doesn't matter if you're DNA or RNA, you can have G, A, and C as your nitrogenous base. But if you're DNA, you can have a T there, but not a U. And if you're RNA, you can have a U as your nitrogenous base, but not a T. Sound good? All right, and a DNA strand, a DNA molecule, is a bunch of DNA nucleotides all linked together, polymerized together. And an RNA strand or RNA molecule is a bunch of RNA nucleotides all linked up together. All right, so let's see. So these represent actually 2 pieces of DNA, 2 DNA strands. And one thing I want to illustrate with this cartoon is that in the cells of living things, you always find DNA in the double-stranded form, meaning you always find DNA where 2 strands of DNA have joined together to form what's called a double strand or double helix. And there's a rule that called the complementary base pairing rules that dictates when two strands of DNA come together, which nitrogenous bases will pair up with each other. And the rule was pretty simple. G and C always go together, and A and T always go together. And I realize I don't have any A's or T's on these two, but if I did, the A on one would always go with a T on the other, and a T on this one would always go with the A on the other. But it does illustrate the GC base pairing rules. But anyway, inside living things, these two pieces of DNA would not be separate. They would always want to come together like this. And that's sometimes called complementary base pairing, or sometimes just base pairing, or sometimes you call it hybridizing, like that. And what's holding them together, does anybody recall? What kind of bond hold these guys to each other? Hydrogen bonds, yes. In the nitrogenous bases, there's lots of partially positive hydrogens and partially negative nitrogens and oxygens, and the orientation of those partially charged atoms perfectly lines up between the G's and the C's, so they're able to perfectly hydrogen bonds to each other. And although I'm not showing it, between A's and T's, there's perfect hydrogen bonding alignment. So that's why A's and T's base pair, and why G's and C's base pair with each other. And what's kind of fascinating is this, that if you were to separate these two back apart from each other again, like that, and in a test tube, also add in a bunch of other pieces of DNA, these two would still find each other. A piece of DNA will almost magically always be able to find its complementary strand and will hybridize to it like this. Now, how is it that that DNA strand will ignore all the other ones? Well, it's just simply that they're not complementary to its sequence. If this strand tried to hybridize with this one, the hydrogen bonds would not be perfect, and so that's why it doesn't do it. The hydrogen bonds would actually push it apart rather than make it stick together. Now, so anyway, inside living things, you always find double-stranded DNA like this. Now, if you were to boil this test tube that had your pieces of DNA inside of it, then you would be able to separate the double-stranded DNA back off again. The explanation is simply that higher temperatures means more kinetic energy, more movement energy of the molecules. They move faster. And if they move fast enough, that provides enough energy to break the hydrogen bonds that were there. So they will separate out if you boil them. But that's not something that takes place inside living things. Living things will die if you try to boil them. And so this is not something that occurs inside real living organisms, but it does occur quite a bit in the laboratory. Molecular biologists is the name of the type of biologist who works with DNA and RNA, and they frequently boil the DNA to separate the strands. If you think about like a mechanic working on your car, the car mechanic has to take the engine apart in order to tinker with it. Well, molecular biologists are all about changing and working with the DNA, and so they have to separate the strands in order to tinker with the DNA.
Is it possible to do any of that here?
Yes. I don't think in our laboratory this year, in this semester, we'll be doing it, but there's another biology instructor, Jeff Huey, and he is actively doing research where he's analyzing DNA and RNA samples, and he has a machine called a PCR cycler, which regularly denatures the DNA, is what it's called. Yes, yeah, definitely. If you're interested in doing some work, I'm not sure if he's still taking students, but I know in the past sometimes he had students come in to do special projects to working with DNA. Alrighty, well, let's see. To summarize a bit where we are, the DNA is the genetic molecule, and it carries the instructions for running the life processes. And this summarizes some of the things we talked about. The sugar in a DNA nucleotide is deoxyribose. They can have A, G, T, and C as their nitrogenous bases, and DNA you always find is a double-stranded molecule because of the complementary base pairing. The RNA nucleic acids are a little bit different. They are single-stranded molecules. Well, maybe I should summarize here. RNA has the ribose sugar in its nucleotides and can have A, G, U, and C as its nitrogenous bases. But notice here, RNAs are not found as double-stranded molecules. They are only found as single-stranded pieces of DNA inside the cell. Here's a picture of an RNA molecule. Notice it has the same backbone that we saw for DNA. Phosphate, ribose, phosphate, ribose, phosphate, ribose, phosphate, ribose. And there the nitrogenous bases sticking out the side. What I'm saying is there's no complementary RNA strand that pairs up with it. You just don't find that inside living things. I think I mentioned last week about the function of RNA. At that time I said DNA is the genetic molecule and carries the genetic information for the cell. And the RNA you can think of as DNA's assistant. It helps the DNA carry out its functions as the genetic molecule. And just to give you an illustration of that, one function of RNA molecules is to be a temporary disposable copy of a DNA molecule. For instance, if you look at the DNA, double-stranded DNA molecule on the left, here you see its nitrogenous bases, T, A, C, G, T, 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 G, like that. And remember what I said earlier, the genetic information in the DNA molecule is written in the sequence of the nitrogenous bases, just like we take letters of the alphabet and can put them together into words and make instructions based on those words, like a cookbook. The order, the sequence of nitrogenous bases in the DNA strand holds the genetic information. Well, look at this RNA molecule over here. It's single-stranded, as we expect RNA to be, but notice it's the same sequence. Instead of TACG, you get UACG, and instead of TTTG, you get UUUG. And just remember that U is essentially the same as T. It's just sort of the RNA version of T. So other than the T's becoming U's, it's the same darn sequence, so it carries the same genetic information. And so in a way, think of this as a photocopy. If you had some original blueprints, some very valuable document that you didn't want to risk being damaged, you might make photocopies of it to work with at your construction site if you were building a building or something like that. And so that's one